So this is a lecture from August 24th, 2020 on the origins and the implications of human longevity. And some of the things we're going to be discussing today are what a complex trait and GWAS are. Also some factors that are involved in establishing and manipulating human longevity. We're going to talk about several major events that helped increase the human lifespan in the 20th century. And then we're going to use the post-lecture questions to talk a little bit more about how the age structure in the United States has changed over the last 100 years in response to these changes in lifespan and life expectancy, and also talk about the societal kind of advantages and disadvantages associated with extending both longevity and health span in humans. And so I just wanted to take a second and review these two different terms because they look and kind of seem similar, but they actually have different meanings. And so I just wanted to make sure we're all on the same page as to what longevity is and what lifespan is. And so take a second and either go back to your notes, pause the video, and take some time to look up what longevity is and then how it differs from lifespan. So what is longevity, what is lifespan, and how are they different? And so take a moment, find those definitions, and then take a look at them. And so longevity is the maximal age potential of a species, whereas lifespan is the mean or the average length an organism or individual is alive. And so longevity applies to a species, right, and not individuals, whereas lifespan applies to the individual and is really just a measure of sort of how long a person lives, whereas longevity addresses like the maximum possible age that a species can reach. And so the book tends to kind of use these interchangeably, but it's important to recognize that they're actually two different things. And so we're going to start by talking about longevity, which is that maximal age potential for a species. And in particular, we're going to talk about it in humans. As we know, and we've talked about previously, humans have a very extended longevity or a very extended maximal potential age. And this originated in a couple of different ways. And so one is a change in the mortality rate. And the mortality rates of humans, or the rates of death, are affected by several different things. And so a decrease in mortality rate has led to extension of human longevity, and that mortality rate decrease has been modulated by the environment. And so environmental factors can make the mortality rates or death rates what are known as facultative or plastic or changeable. And one particular example of how longevity may have been extended by the environment and by a changing mortality rate is by looking at babies that were born in months after the harvest versus before the harvest. And so in historical times, the months after the harvest, so specifically here, September, October, November, and December, people or humans had more access to food and they had better quality food than they did at other parts of the year. And so if a baby was born in these months, they tended to not die and also have a greater fitness. And all of these individuals from the subpopulation were able to pass on their genes to subsequent generations. And so it sort of led to, in humans, this subpopulation of individuals who were all born in these months and that had a greater fitness, passed on their fitter genes, and increased the longevity of the species as a whole. And there are other environmental factors that have affected human mortality rates as well and decreased them. And so by decreasing mortality rates, we extend longevity. And we can decrease in mortality rates can also be affected by genetics. And we know this from some experiments done in Drosophila or fruit flies. And so in this experiment, you can see the mortality rate of a large population of fruit flies, about 1 million of them. And you can imagine that mortality rate over time, or as the animals age, would just be expected to continue increasing and increasing. If it really was just random and subject only to the environment. But what we see here is that in some older populations of flies, the mortality rate actually kind of dwindles down and decreases as those animals get older. And so that decrease late in life in the mortality rate indicates that there's some role of genotypes for longevity or genetic factors contributing, and it's not just purely random and purely linked to the environment. And that the individuals in these populations, these long-lived ones, have some kind of like longevity gene that helps stop them from dying and extends the longevity. And so in humans, we see a similar trend, and these longer-lived subpopulations would have had kind of a greater fitness, and they would pass on their longevity genes, or the ones that suppress their mortality, to their progeny, thereby passing on that idea of longevity extension in the population. So another study that sort of deals with the genetics of changing mortality rates is a study that was conducted in long-lived humans and their siblings. And so this study is specifically dealing with centenarians, which are people who reached 100 years of age, and their siblings or twins. And what was found in this study is that compared to the normal cohort of males and females in 1900, the siblings or the twins of centenarians actually lived significantly longer and therefore had a suppressed death rate compared to their cohorts, right? So you can see the normal death rates for males here in the light blue line, and then below it in the dark blue are the siblings of those centenarians and their death rate, which is decreased in comparison. And so it seems that there is some kind of contribution of genetics, because siblings or twins, people who are related to long-lived humans, also seem to have decreased death rates and decreased mortality as compared to their non-long-lived counterparts, which means there is some contribution of genetics into the process. And so longevity is what's known as a complex trait, or a trait that's derived from many different genes and therefore has highly variable phenotypes. And so sometimes we think of genetics as being one trait that has one phenotype, right? So there's one gene that's, for example, controls eye color, right? And you can have either blue or brown. But a complex trait is much more, as the name suggests, complex, and has sort of these phenotypes that vary in the middle. Height is one complex trait that's easy to think about. Height is derived from multiple different genes interacting and coming together to make those basically a spectrum of different phenotypes. And longevity is similar to that, right? There's a spectrum of different ages that people can live to, and there are multiple genes interacting. And the way that we start to understand what genes those might be is using what's known as a genome-wide association study, or GWAS. GWAS allows us to study many different complex traits, like aging and longevity, and also like some genetic diseases. And the way that GWAS works is basically by putting people, in this case humans, into two separate groups. One that has whatever trait you're interested in studying, and one that doesn't. And so in the case of longevity, we have the cases being humans that are centenarians, right, that live to 100 years old, or old, the maximally old humans, and we compare them to a control group of just normal aging people. And then what we do is we sequence the entire genome of both of these groups. And so this is pretty cheap at this point to sequence the genome of a human. And then we compare all of the genomes of those old or long-lived people with the genes of those in the normal aging group. And we look for differences between the two of them. So we look for differences between the general population and those who live a long time. And we do that by looking in specific spots in the genome that have what are known as SNPs, or single nucleotide polymorphisms. And these SNPs are at particular locations in the genome. And so we can identify the variance between long-lived and humans in the general population by looking first at the SNPs. And then we can start to make associations from that. So if we know where the SNPs are, and we look for differences in SNPs between our cases and our controls, and we find out that there's a lot of differences here, for example, we can then make some associations between the SNPs that controls have and the SNPs that cases have, and find areas on the chromosome that might be involved in aging. And what the output of a GWAS study is something that looks like this. And so this is a map of all of the chromosomes, 1 through 22, laid out here. And the SNPs are all of these individual dots. And so the SNPs serve as kind of a guide, or so we can map other genes involved. And so you can see here that there are a lot of different SNPs. But below this kind of dotted line, or this threshold, the SNPs are the same between the cases and the controls, or the long-lived people and the general population. Anything that reaches above this line would indicate a SNP or a variant between these two groups that might be involved in longevity. Right, and so we can see here in this purple chromosome, there's a couple SNPs that might make the cut. There are some on this blue chromosome, or chromosome 19 as well. And so by looking at these differences in SNPs, seeing which parts of the genome vary in our long-lived people, we can start to look for other genes in that same location on the chromosome and sort of pick out the ones that are involved in aging. And so GWAS can't actually tell us what these genes are directly, but it can tell us where we should start looking for those genes. And then this is an additional GWAS study that was published in PLOS Genetics in 2015 that was looking for longevity genes in humans. And it actually identified 8 different loci, or locations, where these genes might be located. And so it's not quite as beautiful, but this is the same Manhattan plot, or this plot of SNPs from chromosome 1 to 22. And these are just 2 different cohorts of long-lived people. And what they found in this study was that there are 8 different loci, or 8 different genes, that are located in these specific locations of the SNPs that might be contributing to longevity in humans. And so they compared long-lived people to the general population. They found these places where there were a lot of SNPs, or a lot of differences between the 2 genomes. They started looking at what else is there, and then they came up with these genes, which you can see listed here. The one that has the highest correlation, or the one that seems to have the strongest potential to be an actual longevity extending gene, is what's known as ApoE, or apolipoprotein E. It's a protein involved in lipid metabolism, and it's actually highly correlated with Alzheimer's disease as well. And so there have been a lot of kind of subsequent studies after this one looking into the role of ApoE and how it's extending longevity and lifespan in humans.